Synthesis and analysis of antimicrobial properties of isoxazoline. I abstract a simple procedure for the synthesis of isoxazole derivatives was developed. Cyclocondensation reaction of chalcones with hydroxylamine hydrochloride catalyzed by an acid produced isoxazoles. Chalcones were synthesized by the condensation product of acetophenone in combination with aromatic aldehydes in presence of strong base. The chalcone synthesized by base catalyzed condensation of 3-acetyl-6-methyl-2-H-pyran 2,4-3H, dione, DHA, with different aromatic aldehyde in good yields. These chalcones were used for synthesis of derivatives i.e. isoxazoline. The synthesized new compounds were characterized by spectral studies i.e. IR, 1H-NMR, and mass spectral analysis and elemental analysis, before being were screened in vitro for their antimicrobial susceptibilities against different bacteria and fungi species. The derivatives were further used for the estimation of its biological properties. From the study it was found that the synthesized compounds are efficient for further research work. Two keywords: dihydroacetic acid, DHA, chalcone, 3-cinnamyl 4-hydroxy-6-methyl-2-pyrones, IR, 1H-NMR, antibacterial activity, antifungal activity, isoxazoline. 3. Introduction. Isoxazoline is a 5-member heterocyclic compounds containing N and O atoms adjacent to each others. The dihydric derivative of isoxazoline are also called as isoxazole. The compound containing N functionality, C equals NO, undergo cycloaddition reaction with an SS unsaturated carbonyl compound or with an alkane to produce isoxazoline having an NO moiety. O being next to N rises the barrier to N inversion to such extent that a temperature lower than the ambient individual can be identified by 1H-NMR spectroscopy 1-2. Synthesis of isoxazoline derivatives has been a subject of consistent interest because of the wide applications of such heterocyclic in pharmaceutical as well as agrochemical industry. 3. Numerous compounds containing isoxazole, pyrrosole, and pyrimidine moiety have been reported as active hypoglycemic, antidiabetic, antipyretics, analgesics, anti-inflammatory, antiviral, antiallergic, anti-cancer agent, antidepressant and antimicrobial agents which include antibacterial sulfonamides, semi-synthetic penicillins, and cephalosporins. 4-6. These discoveries led the scientists to attempt the synthesis of isoxazole, pyrrosole, and pyrimidine derivatives from the chalcones made from aromatic aldehydes and aromatic ketones and to evaluate the synthesized compounds against antimicrobial, antifungal and antitubercular activity. 7-10. The reactive intermediate chalcones involved in their synthesis also exhibit wide range of biological activities. 11. These properties were used in the research to find some novel compounds for pharmaceutical and agricultural industry. 4. Methodology. Synthesis of substituted isoxazoline. A mixture of DHA chalcone, 0.001 mol, and hydroxylamine hydrochloride, 0.0015 mol, and sodium acetate, 0.002 mol, were refluxed for 6-7 hr in 15 ml ethanol. After completion of the reaction, checked by TLC. The reaction mixture was cooled and poured on ice-cold water. The separated solid product was filtered and further washed with cold water, dried and further recrystallized from ethyl alcohol. The structures of isoxazolines were confirmed by spectral analysis, IR, 1H-NMR, mass and elemental analysis, 12-13. Analytical characterizations. The IR spectra of compound were recorded on FTIR spectrometer, 1H-NMR Avance 500 MHz, CDCL3, instrument using TMS as an internal standard, chemical shift are given in dppm. The mass spectra recorded on Shimadza spectrometer. IR spectra. The IR spectra showed characteristics absorption band at 1610-1640 cm1 due to the C equals N stretching. A broad peak showed in between 3000-3500 cm1 is due to the hydroxyl group in synthesized compounds. 1H-NMR spectra. 1-HNMR of isoxazoline showed following type of peak, which confirms the formation of product D3.0 to 3.8, DD, 2H, CH2, i.e. Ha and HB of isoxazoline ring. D5.0 to 5.4, T1H, CH, i.e. HX of isoxazoline ring. D14.2 to 16.0, S, 1H, O, while the ring peak at D2.15 to 2.8, S, 3H, ARCH3, all such peaks are observed. Mass spectra. Mass spectra of representative compounds confirmed the molecular formula weight of respective compounds. C:\users /JCL850301/desktop/IAO.jpg. Fig2 synthesis of 3-4-hydroxy6-methyl-2-azo-2-H-pyrin-3-YL-5- substituted aryl-4,5-dihydro-2-isoxazoline, MBOI-MBOV. Synthesis of 3-4-hydroxy6-methyl-2-azo-2-H-pyrin-3-YL-4,5-dihydro-2-isoxazoline, MBOIV. A mixture of 1-4-hydroxy6-methyl-2-azo-2-H-pyrin-3-YL-3-2-fluorophenyl-2-propenon, 0.001 mole, hydroxylamine hydrochloride, 0.0015 mole, and sodium acetate, 0.002 mole, in 15 ml ethanol was refluxed for 6 to 7 hr. After completion of the reaction checked by TLC, the reaction mixture was cooled and poured on ice-cold water. The separated solid product was filtered, washed with cold water, dried and recrystallized from ethanol to give the corresponding product 3-4-hydroxy6-methyl-2-azo-2-H-pyrin-3-YL-4,5-dihydro-2-isoxazoline. Similarly, all the other compounds of the series also prepared by the same procedure. The yield and MP are listed in Table 1. For biological activity. Preparation of media plates for maintenance of bacteria and fungus. Nutrient agar, 28 gm/1000 ml, was dissolved in 1 lit conical flasks and sterilized in autoclave at 1210 C, 15 lbs/square inches, for 20 minutes. Potato dextrose agar, 39 gm/lit, was dissolved in 1 lit conical flasks and sterilized in autoclave at 1210 C, 15 lbs/square inches, for 20 minutes. After cooling, medium was poured in sterilized Petri plate about 25 ml. The media plates were inoculated aseptically with inoculums. The bacterial culture of S. aureus, E. coli, Salmonella typhi were inoculated on nutrient agar medium. The bacterial culture plates were incubated for 24 hours in biological oxygen demand, BOD, incubator at 280 C temperature. Growth of organism. The fungus culture plates were inoculated on potato dextrose agar medium. The fungal culture of Fusarium oxysporum, Candida albicans, and Aspergillus flavus plates were incubated at 35 to 370 C temperature in BOD incubator. Growth of organism 14-16. Antimicrobial activity of compounds by agar well diffusion method. Prepared media plate which was not inoculated with pathogen was taken and the wells were bored into agar using a sterile 6 mm diameter cork borer. 6 bore was made in each plate. The culture of the test organism was spread on Petri plate with sterilized spreader chalcone derivatives of desired concentration. The test compound, 250 g/ml, was dissolved in dimethyl sulfoxide and loaded on a sterile filter paper disc of 6 mm diameter. The Petri plates containing nutrient agar medium were spread with 100 L of actively growing both culture of the test bacteria using sterile cotton swab and allowed to dry for 10 minutes. For fungal species, 100 L of active culture was spreaded on PDA. Then the impregnated discs were placed on the surface of inoculated agar medium. With respect to control, DMSO is used as a control for testing antimicrobial activity 17-18. The positive control was taken as chloramphenicol. The plates were allowed to incubate at room temperature for about 2 HR and then plate were transferred to incubator at 28 degrees Celsius for bacterial culture and 35 to 37 degrees Celsius for fungal culture. After 3 to 4 days, zone of inhibitions were examined and recorded. The experiments were performed in replication and average diameters of the zones of inhibition were calculated and mentioned in Table 2. The observation shows that both the substituted chalcone derivatives are positively having antimicrobial property in action. It seems to be bacterial and fungicidal activity in them. So, they can be used against different animal and plant pathogens to cure a disease as a protection agent. 19-20. V. Result and discussion. A series of novel substituted isoxazoline were synthesized by refluxing a mixture of DHA chalcone, MbCl-MbCv, hydroxylamine hydrochloride, and sodium acetate in the presence of ethanol. The reaction is completed within 6 to 7 HR as monitored by TLC, providing good to excellent yield 75 to 80%, MBOI-MBOV. All the synthesized substituted isoxazoline derivatives were confirmed on the basis of spectral analytical data, IR, 1HNMR, mass and elemental analysis. The IR spectra of the isoxazoline derivatives showed disappearance of C=O bond of chalcone. A strong band appears at 1590-1615 cm1 assigned to C=N because of ring closure. The absorption band 1450-1465 cm1 due to CH2 of isoxazoline ring present in the respective compounds. 1HNMR spectra of some representative compound were recorded which show the characteristic peaks at D3.0-3.6, DD, 2H, CH2IE and HB of isoxazoline ring, D5.25.4, T, 1H, CHIEHX of isoxazoline ring, were observed. All the other aromatic and aliphatic proton were observed at the expected region. The antimicrobial activities were tested against the bacterial and fungal species as mentioned in methodology. The effective zone of inhibition were observed at each concentrations mentioned in Table 2. All the isoxazoline compounds are having effective antimicrobial properties as shown in Fig 2. The results were compared with standard chloramphenicol and the positive control was taken as DMSO. There was no zone of inhibition in DMSO on microbial growth. Both the organisms, i.e. bacteria and fungi, showed maximum zone of inhibition, i.e. 23 mm and 21 mm, respectively as shown in Table 2. In conclusion, the synthesized isoxazoline compound possesses a good to moderate biological properties. These compounds will be having application in pharmaceutical, agriculture, medical field for drug development.